So the lecture today is part of a two-lecture series in cytometry meant to be an introduction for new users. It's slightly basic at the same time. It's a little more. It's a little in depth in a way for an intro lecture, but I think it's important because providing all these details will help you to understand the technology, the capabilities, but also the limitations. The first lecture will be covering principles of instrument design, instruments design that is shared across different instruments in the platform. Specifically, I will be going through the path the cells go through as they get analyzed by a cytometer, including fluidics, which is extremely important, and it's a subsystem that you guys will be dealing with as you run samples in the instrument. Then I will be covering the signals and the optics with a focus on light scatter and fluorescence, which are the main measurements that cytometers do. I will be briefly describing electronic principles, how the pulses are processed by the cytometer, and including key elements that are important for you guys as you set up instrument experiments for the analyzers or the sorters, which are acquisition triggers and thresholds. What do they mean? Properties of the pulse that the instruments store and measure. A brief intro into spillover per compensation and full spectrum unmixing. Then I will be describing basic principles of data in cytometry and how this data is visualized, some of the limitations and artifacts that you might encounter, specifically talking about limit of detection, which I think is something that is very rarely covered in cytometry, but in my view is one of the most important things to take into account before you plan an experiment for a cytometer. And I will end with a list of applications, examples of applications that I think are important for everything that is starting, because it gives you an idea of the technology capabilities and how can you use cytometry to answer research questions. Now, the second lecture will be on Friday, and the second one will be a little more practical. So I will be talking about things including how to set up an experiment for cytometry, how do you, the information you need to know before you start, how do you prepare your samples, how do you set up the instrument, how compensation happens, and all the controls that you need for a proper compensation. I will be describing panel design, which in itself could be a different lecture, but it's an important thing in order to make sure that your panels and your experiments are successful. Going through experimental controls and examples of data acquisition analysis, including again artifacts in these topics, and I will finish with cell sorting. So as a bit of introduction into the cytometry platform, I prepared a series of slides showing you where we are, what we do, how can you reach us. The Melbourne Cytometry Platform is one of the largest equipment-based platforms at the University of Melbourne. There are several nodes for the platform. They are all within the university, but across different schools, departments, and faculties, and mostly are located around the Parkville area. So the bigger node, the biggest node in the platform is the Peter Doherty Institute, with a lot of instruments, a lot of users, and staff. Melbourne Brain Center is the second largest node, followed by Bio21. We also have the dental school, the Faculty of Veterinary and Agricultural Sciences. And over the years, the nodes have been changing dynamically, so some nodes have been absorbed by other nodes. Last year, with the fusion of pharmacology and biochemistry departments, we got now a bigger node in Bio21 with 3 instruments, but essentially it's a dynamic platform. What we do several things. One of the things is to manage the resources and make sure the instruments are apt to the challenges and they are perfectly performing, that we have instruments that are updated, that we can negotiate purchases and essentially join forces toward application for grants and things like that. We also manage the booking system, which is a unified booking system that covers all the resources that we have in the platform. We do a lot of inter-node staff support, so we typically work with each other, trying to solve problems and help users, assist nodes that are not staffed. We also deal with the user training and the collaborations with users for high-end applications. The platform stack can assist the use of instrumentation, specifically when users are starting or if they are doing cell sorting. These are applications and services that are mostly all the time provided via systems by a staff. But most of the instruments that we have can be, after induction and training, used directly by the users without any supervision. We also are the primary contact for anything related with instrument capabilities, experimental design, applications, and data interpretation. As I mentioned, we also work towards purchases and trying to keep the equipment that we have up to date. And a lot of things that we do also have to do with characterization of instruments. We want to make sure that they are fine. That means we need to know elements of QC and QA to make sure the instruments are in optimal condition. And at the same time, we also manage some of the software licenses that you will be able to access once you become a user, and you need to access the license at a fair price to analyze your data. Now, I'm including here main contacts for the platform. We have all the staff that works in the platform across different nodes. I will be the primary contact for anything related to the dental school node, but also you can call me and send me an email for anything related to platform general issues or inquiries. Vanta Jameson is the manager of the brain center node. Lydia works as an operator. She just started recently. Tina Liluk is the Doherty node manager. And Catherine, Angela, and Lankesha are operators there, supervising and taking care of the instrumentation and running the sorters. Elizabeth Ann Washington is the primary contact for the FEVAS node. And if you have any questions, you can always, of course, contact us for general inquiries via the email that we have, Melbourne Cytometry. Now, the Cytometry platform have many instruments, around 30 instruments in total. These numbers keep evolving as we retire old instruments and buy new ones, but they represent a broad variety of resources and capabilities. So we have, for instance, the cell sorters all at the top, and different brands of analyzers that have different performance, different characteristics, and bring different things to the platform and for the users. For test analyzers are found in most of the nodes. It's a very highly used instrument in the platform. It's also one of the oldest instruments that we have still. And some of the instruments that we have are not within the platform. So the CyTOF and the MCP screen is available via the materials and characterization and fabrication platform. Now, the instruments that the cytometry platform have differ in terms of what they do, or which lasers they have, and which fluorochromes they can detect. So I will be classifying this in terms of analyzers only. So you can run samples, but you cannot isolate samples and cells with these instruments, which differ in terms of the lasers that they have. So different instruments will have different laser configurations. And of course, that means you can excite different fluorochromes all the way from UV to infrared. Most of the instruments have, in addition to the capacity to analyze cells by tube, you can also run cells by plate, so you can do high throughput screening. We have the sorters, which are a specific type of cytometer that have all the analytical features of an analyzer, coupled to the ability to sort and isolate cells and populations. The instruments that are in the MCP platform, they also provide high dimension analysis. So in the case of the CyTOF, it's using mass spectrometry to measure over 50 markers. we also have in the platform, in the cytometry platform, instruments that can reach at this point 45 different markers with the Aurora spectral analyzers. And of course, you have also the imaging cytometry side of things, which is a combination of cytometry and microscopy, so you can run cells, do raw measurements as you do in cytometry, but also capture images that give you an idea of fluorescence location. Now, over the years, we have been characterizing the instruments that we have, and we know that the recent purchases that we have done, for instance, the Aurora or the Cytoflex, have brought different virtues and qualities to the services and the performance and capabilities that the platform have, including things like sensitivity, high dimensional analysis capabilities, the ability to extract autofluorescence from measurements, so make the measurements more precise. So if you have any question concerning which instrument to use, my advice is to contact us. All the instruments in the platform are available to all the users. The reason why we have this difference in instruments, and sometimes the instruments are located in different nodes, is because instruments purchases tend to match the needs of the node. So if in MBC people are working with nanoparticles and small particles and high sensitivity, etc., and dim signals, well, you need to match this with instruments that can cope with the detection limits requirements. In the case of Peter Doherty, where most of the users are immunologists, you need to have high dimensional analysis and support for it. So that's the reason for the Auroras. But any instrument can be accessed by any user after induction and training. It doesn't matter if you are from the university or if you're an external user. Once you get training, you get access to our booking system, and you will be able to place bookings in any instrument in the platform via our Calpendo calendar. Now, for additional information on the platform, my suggestion is to check this webpage that we launched last year. You'll see elements of capabilities, details on access, news and events. For instance, this current series of seminars are advertised in the platform, in the website, and we will have advertisement of incoming lectures and seminars organized by us. Description of who we are and the resources that we have. Now, for most of the lecture, I will be talking about what is a cytometer and what are the elements that make a cytometer, including elements of fluidics, electronics, and optics. And although it looks like maybe too much information, I think it's essential because most instruments at this point are enclosed. You cannot see the elements, and issues that you can have with the instrument can be related with any of the subsystems. So knowing how the instrument operates inside the box is important to make sure that you get the best out of the cytometry techniques. Now, we could say that cytometry is one of the oldest omics, and by that I refer that cytometry is over 30 years old, but since the beginning it was extremely powerful in providing a tool for the streamlining of the analysis of cells in single cell suspensions. So it is super fast. I mean, over the year, the electronics have been handling and been enabling cytometry to go faster and faster in terms of how many events per second a cytometer can process. But I would say that roughly it will go between 30,000 to 100,000 events per second at this point, which means that you can very quickly identify rare populations, and you can very precisely do correlation between parameters and markers. So imagine 1 million cells under these speeds and rates will be acquiring something like 10 to 30 seconds. So this is very quick technique. The other thing is that cytometry provides measurements in a high dimensional level. So you measure not only one property, but several properties at once out of each individual cell. And you can resolve different subsets out of complex mixtures, and you can essentially do correlation between imagined treatment of the animals and the evolution of cytometry subsets in the immunological system, etc. You can do all this level of expression, timing, kinetics. It's quite powerful in terms of the information that it provides. The other important thing is that since you're analyzing so many cells, you get a pretty fair idea of statistical robustness. So when you're saying the percentage of expression is this, You are confident in the numbers because you have acquired enough cells to trust the statistical outcome. And the other thing that is very important in cytometry, which makes the technique still relevant today, is the fastest way to isolate all these populations, these specific populations based on the properties measured above, and get pure or bulk single cell isolations that are important for as a necessary step before other applications like advanced fluorescence microscopy, sequencing, proteomics, metabolomics, you name it. Now, cytometers can be benchtop analyzers, or they can be high-speed cell sorters. I'm giving you examples here of instruments that are currently in the platform, and you can see they look quite different, and some of them tend to resemble a black box, white box in this case, but essentially they are enclosed and kind of inaccessible. Sorters tend to be a little more advanced in terms of the electronic components, the fluidic components. They are meant to run for hours and be able to isolate populations. So yeah, the complexity will be higher than instruments like analyzers. Analyzers can come coupled with high throughput units, options that allow you to run, for instance, cells from plates. So you can essentially run your plates and go to your lab and the acquisition will be happening. But in spite of all the difference in appearance, instruments tend to follow the same general structure. So what is the structure, the general structure of a cytometer? And it's all based on what the instrument can do. So cytometers take particles in suspension, so this is a requirement. Only particles in suspension can be processed by a cytometer, and they might make sure that the cells travel in a single file and at constant speed across different illuminated volumes, which in this case are provided by lasers, high power lasers, where scatter light and fluorescence is collected. So the whole point of the initial part of it, which is handled by the Fluidics, is to guarantee that single cell suspensions are essentially focused into a single line of particles that are consistently, in terms of speed and position, running across lasers, producing consistent and compatible scatter and fluorescent measurements depending on their properties. Yeah. Now after that, this light needs to be collected by the optics, so there are different optical configurations, and the final fate of the photons generated will be photon detectors. Then you have the electronics that will convert all these signals into data that can be analyzed in plots. Yeah. A sorter have all the properties, so all the fluidics and optical components, and on top of that you have the sorting elements, which handles electronics and the robotics that would allow you to isolate populations and into specific vessels. Yeah. Now this is an expanded view of the slide that I showed you before. I showed you essentially schematics of the principles of the cytometer design. So you have the fluidic subsystem and the flow cell, where the cells are focused. You have the laser sources. You have the optical filters and detectors. How the signal look like in terms of nature is essentially you're measuring pulses, and how it's all converted into electronics. But in the process, you have several subsystems. You have the fluidics, which have to do with anything related with sample delivery, velocity and consistency, velocity and flow. Fluidics work primarily with laminar flow. Essentially, laminar flow is the main principle of operation of fluidics to guarantee these consistencies, and also via hydrodynamic focusing. So I will be explaining in the next slides what this means. You have the optics that include anything related with laser, focusing lenses, acquisition and collecting lenses, pinholes, optical fibers. Everything has to do with the excitation and focusing of the laser, collection of lights, and delivery of light into detectors. You have electronics, which are the controls of all the subsystems, and you have different things that amplification of the pulses, analog to digital converters, pulse processing. You have the computer, of course, and you have a crosstalk between the computer and the software and the instrument at all times. And then in sorting, you have different things which are more specialized. I will be covering this tomorrow, but essentially include things like jet formation, droplet generation, coincidence analysis to guarantee purity, droplet charging, and the harvesting elements. Now, important thing in cytometry is that although we can divide the subsystem into different classes, it is important to understand that they all work together and they all collaborate towards a final aim, which is to be able to measure properties out of a single cell in a consistent manner. Now I'm showing you here an example of the fluidic components. I will be focusing on fluidics in the first section of the description of what the instrument does. And the reason for that is fluidics is utmost important. As I mentioned, you need to measure things precisely. You need to be able to measure fluorescence and scatter of particles, but in a way that all particles have an even chance to be able to produce photons that are related to the internal properties, not the velocity or the time of exposure or the position in which I cross the laser. So you need to be consistent. And the fluidic components handle that. So you have, if we can go in direction from when the sample is placed, so you have the sample in this case, there are different valves that will take the samples and the cells into the flow chamber, which is the heart of the cytometer, where everything happens, where the particles are focused and where the particles meet the laser. You also have sheath tank. Sheath tank, essentially the instrument is always, once you turn on the instrument, always running some liquid. It could be saline, it can be water, it can be PBS, depending on the instrument. And also, of course, the waste management system. So you will have then pressure valve that would allow you to control the pressure in the sample compared to the pressure in the sheath that would allow essentially to inject samples into the running sheath, into the instrument. This is a very, very simple schematic of how fluidics works. And of course, they are different between instruments, and depending on the design, the level of complexity will change. But something that I want to bring your attention to is that although it seems simple in terms of overall description, there are many, many, many elements to take into account. And I'm showing you here an example of probably the first successful benchtop analyzer ever made, which is the Faxcalibur. We used to have one of those, but it's no longer operational. But you can see how many elements in terms of electronic design and components are involved into guaranteeing that the measurements are consistent and the velocity is constant and the laminar flow is preserved, etc. Yeah, so it's a very complex system. But the most important thing to take into account, to understand when you're thinking about fluidics, is that fluidics require laminar flow. Now I'm presenting here examples of laminar flow and turbulent flow and how these flows affect the position and the flow of particles like erythrocytes. So you can see turbulent flow, which is stochastic, unpredictable, with sudden changes in variations of pressure and velocity, distort the erythrocyte measurement and essentially kind of produce some kind of random flow that will never be consistent. Yeah, laminar flow, in contrast, produce streamlined, beautifully defined, predictable flow of particles that you can anticipate their position anywhere in the system. And some of the properties of the laminar flow, well, it could be the parabolic nature of it, the laminarity of it, the fact that layers will never mix. So that means that when you inject particles in a buffer like, I don't know, PBS, they will remain in that PBS compartment and they will essentially be constricted and carried by the sheath flow, as I'm showing you in this slide. The most important thing out of this slide is that the only way cytometers can guarantee this consistency in terms of measurement is if laminar flow is in place. Now I'm showing you here an example of what happened, one of the interesting outcomes of laminar flow. So this is a video that I found in YouTube that have 2 million views. It's a kind of instructional video. It looks like a video done for a school or something, but what they have is they have injected ink corn syrup into a high viscosity glycerol container. There is a cylinder in the middle, and what they're doing at this point is they are essentially moving in a kind of constant fashion and a slow speed. The inner cylinder will produce some kind of friction between layers of glycerol that drags all the way into the corn syrup that have been injected. So they're doing like 7 turns of these, and you see that the effect appears like this ink is becoming suddenly like a big layer, and that layer essentially have extended, and you essentially cannot distinguish a different ink that you have injected. Now, something that they do or makes the instrument, the video interesting, is that they go back now. After 7 turns, they will go back to essentially try to do the 7 turns back at the same kind of velocity, and what will happen at the end will show you something that makes laminar flow so important, is that laminar flow is predictable. It means that the flow of particles in laminar flow can be anticipated, can be predicted. So if you inject particles at this point in the flow stream, you can anticipate the position, imagine, a few centimeters away from the injection point. And that is why laminar flow is so important. So I'm giving you here 2 examples of fluids, the different viscosity. You have honey, you have water, and you can see that both liquids can, in this case, in the examples that I'm showing you here, run in a way that seems to suggest laminar flow. In the case of honey, because of the viscosity. In the case of water, because of the design of an engine that will produce laminar jets. So you can make liquids with low viscosity, like air or water, to flow in a laminar fashion. And other liquids like honey that are highly viscous will tend to naturally flow in a laminar fashion. Like, what essentially defines the movement of a liquid? So how can you guarantee that an instrument is running on the laminar flow? And the example that I'm showing you here, I mean, I'm showing you examples of the history of how this was discovered, what are the elements that are involved in the definition of fluids flowing as laminar systems or turbulent systems. And most of this knowledge was part of the work of Osborne Reynolds in the 19th century. He was working on experimental setup to try to play with different parameters to measure and see essentially the fate of the ink that have been injected into the flowing container. So he observed cases of laminar flow in some conditions, or transition from laminar to turbulence. Turbulence will have different shapes depending on the parameters he was using. And one interesting case is alternating laminar and turbulent flow with the same pipe across the trajectory. So you can have laminar turning into turbulent, then resuming back to laminar, etc. This is what you're looking at here, is the fate of the ink that he's injecting. Now, he discovered that you can describe the outcome of a flow, either in terms of laminar or turbulence, be a number that later on was known as the Reynolds number. And it's a number that is adimensional, but essentially contains all the elements that define how the flow will be. So one of the elements that are important is the velocity of the fluid. So the higher the velocity, the higher the number that you get. The diameter of the pipe was another important factor, how big this internal cross-section was. The density of the fluid and the dynamic viscosity of it. And he essentially defined that when the numbers are under 2,300, the system will be mostly laminar. Transient flow, cases like this, in which you will have transition between laminar and turbulence, will happen when the numbers are between 2,300 and 4,000. And turbulent will most likely happen whenever the Reynolds number is over 4,000. Now, the good thing to remember here is that, and to know, is that cytometers have been designed to maintain the Reynolds numbers under 2,300. So by playing with the dimension of the pipe, the velocity of the fluid, the pressure that you apply to the system, the instrument should be quite robust in terms of maintaining and be able to resume laminar flow conditions. But something you need to know is that you can distort laminar flow big time. And this happens, for instance, when you run samples that are poorly prepared. If you haven't filtered your cells and you have large aggregates, well, these aggregates and viscosity that arises for things like cell death and DNA release could affect your laminar flow and disrupt it. So your measurements will stop being consistent in those cases. The other thing that is very important is sometimes you can introduce air bubbles in the system. For instance, by leaving the instrument dry, which is something that we have seen quite frequently, specifically in nodes that are not directly supervised by staff. So air bubbles, air in the system is enemy of the laminar flow conditions. It distorts the velocity and changes the quality of your measurements. The quality of your sample is super important, and it's something you always need to keep in mind. Filter your cells, always. As long as you can see any kind of aggregates or kind of suspended particles that are bigger and visible to the bare eyes, well, this sample need to be filtered. Otherwise, you will get to things like blockage, turbulence, and imprecise measurements. Now, why laminar flow? The reason why it's called laminar is because when you consider, for instance, the fate of the ink that I showed you before in the experiments or Reynolds experiments, it almost feels like the ink is traveling on a separate layer from the rest. And it behaves a little bit like a bunch of cards that you have essentially spread out. You see that each card is independent and they have some level of friction between them, but you can separate them. The same thing happening in laminar flow. The other interesting thing about laminar flow systems is that over a period of time, laminar flow tend to form a kind of parabolic velocity profile, in which the velocity in the center is twice the average velocity of liquid across the whole pipe. And that difference in velocity that go all the way from zero near the pipe walls all the way to the center creates some interesting outcomes. The first thing will be a gradient of pressure. And this has to do with the Bernoulli's principle and the idea that fluids that are running at high velocity have an intrinsic lower pressure, which is something that is counterintuitive, but is extremely important and have been, for instance, enable us to create aircrafts. And the idea that the velocity of the liquid, the velocity of the air in this case, that's a fluid, is much faster at the top of the wings of the plane that is underneath, and that difference in velocity between the top and the bottom create a lift that can essentially raise the plane up into the sky. So that's how important these gradients in velocity with the impact on gradients in pressure impact. But in the case of cytometry, the practical application of this is once you inject particles in a flowing stream, they will rarely touch the walls. And they will tend to slowly and gradually locate around the center where the velocity is maximum and the pressure is minimum. So you get some sort of alignment just by the fact that you're running things on laminar flow. Now the heart of the fluidic system is the flow chamber. And the flow chamber is specially designed to comply with several properties, including hydrodynamical, optical, mechanical, and electrical characteristics. So particles are typically injected into the flow right very close to the flow chamber or within the flow chamber. And within the flow chamber, things like hydrodynamic focusing ensure that particles are aligned. It works a little bit like a lens focusing light. So the same way you use lenses to focus light, you could imagine that the flow chamber have structures that would allow the focusing of liquids, specifically the core where the particles are traveling into a single file. It would also help with the orientation of particles. So in cytometry, particles tend to run through the laser in a kind of precise, seemingly consistent orientation. And of course, it will guarantee the speed of the measurement as well because it accelerates the flow. But also, flow chambers, in the case of cell sorters, are crucial in the definition of the quality of the liquid jets that are used for the isolation of droplets. And of course, the consistency and velocity that will guarantee the consistency and frequency of droplet generation and things like that. Now, there are two kinds of flow chambers. One is what is called jet in air, in which the particles jump into the jet, into the air, and the laser meet the particles in the jet that is falling. So this is called jet in air configuration because the measurements are done in the air on the jet that is falling out of the flow chamber. In the case of the analyzers, well, the interrogation happens within the flow cell itself. These are quartz cuvettes that we have in Fortezas and other instruments that the laser essentially travel across the quartz, meet the particles as they're traveling inside the flow cell, and the measurements are produced. Yeah? Now, these are two examples that we have in the platform already of what the cuvette instruments look like. In the case of the Fortesa, you see the quartz elements and you see the laser interrogating the particles inside the cuvette, and the different detectors then align to collect the light that is produced out of this encounter. In the case of the MoFlow Astrios and a MoFlow XDP, well, the interrogation happens in the air, and a jet that is falling from the tip of the nozzle in this case. And although you cannot see the interrogation much, you can see the patterns of diffraction that happens when the laser meets jets. And although it's kind of obscured in this case, well, collecting lenses will be in the back, collecting all the fluorescence, and the scatter will be collected to the side. Now, one of the things that happen in the flow chamber, which is important, is the fluids accelerate. And the idea, the way they accelerate is via the use of a jet. Essentially, it's a pipe that decreases the cross-sectional area quite substantially. For instance, in the case of the MoFlow XDP and Astrios, you get a nozzle. Typically, will have a 5 millimeter at the entry point and something between 100 microns or 70 micron output orifice. Now, what happens when you inject liquids in a very large cross-sectional area at a given velocity and attempt to pass it through a smaller area is that the fluids will accelerate substantially. And it can be so strong that, for instance, in the jet in air instrument, the jet is flowing at a rate of 10 to 20 meters per second. And this difference in velocity and this acceleration of the particles have different outcomes in terms of alignment and velocity and single filing. And I attempted to summarize this in this slide, in which I can show you, for instance, that since there is acceleration within the nozzle, particles that are a little bit up front but barely separated from each other, these particles that are up front will have always a higher velocity across the nozzle, which means that they progressively will separate from each other. So things that are very close in suspension eventually become a single file with a nice gap in between, which allows measurement of single individual particles as they cross the laser. So first outcome of the acceleration of the fluid in the nozzle is a single filing. The second thing is alignment. For the same reason, so particles that are long and they are connected, in this case I'm putting the same two particles but connected with a kind of bridge. The ones that are up front tend to flow quickly, which means that the pressure and the balance of pressure will make the particle to align and become some kind of a longitudinal alignment kind of flow pattern that will make the measurement consistent across particles of the same type. The last possible outcome for particles that are extremely large, as I mentioned, there is a massive gradient of velocity across the nozzle, which means the pressures also will change. If the particle is too large and the conditions of the flow are too extreme, particles like neurons, for instance, could break apart, which is something that is taken into consideration, for instance, for sorting. So we know this, and the pressures and the systems that we use and the setup that we use for large particles is quite different to what we applied, for instance, for bacteria or lymphocytes. Now, something that is important to take into account, because it's something that you will have access to, is the sample sheath differential pressure. So in order to get the particles to flow, a pressure in some instruments is applied to the sample tube. So that pressure can be higher and needs to be higher than the sheath pressure that is running inside the instrument in order for the particles to be injected into the system. But depending on the differential pressure that you apply, how much pressure you put in the sample, you can go for very little pressure to a very defined path of injection, in which cells will be essentially traveling across a very tiny core. And this means typically a higher precision in the measurement at the end when it crosses the laser. As you increase the pressure, essentially progressively the core will enlarge, and sometimes will be bigger than the output orifice of the injecting needle that you have in the system. So taking care of the pressure is important in order to guarantee consistency in the measurement. And although you would like to run imaging faster by increasing the pressure, yeah, your measurements will also be a little compromised. It depends on what you are measuring as well. But my suggestion would always to use the minimum pressure that you can afford. In the case of nanoparticles, for instance, you need to use that because it's a mandatory requirement for things that I will explain later. But differential pressure is important. High precise measurements or linear measurements like DNA content, for instance, are typically measured with a low differential pressure to guarantee a tight peaks and a very nice precision. Yeah. Now, we have aligned the particles, we have also accelerated the particles, and they have been traveling now through a very tiny core. So what happened then? The particle arrive and meets first laser. Typically, lasers in the cytometers are essentially spatially separated from each other. So imagine it would encounter the first laser, they would encounter the second laser, and after hitting the laser, you will get light scatter and fluorescence produced. Now, something that is important to take into account is that the velocity of the flow dictates the time of exposure. So, since everything is defined by the design of the instrument and the velocity have been is also conditioned by the design of the flow cell, the exposure time in cytometry is a fixed value, and typically can be from a few milliseconds to a few microseconds. This is very little time to get enough signals of particles, you would think, which explains why in cytometry we typically use high powerful lasers. So we cannot afford to use lasers or light sources of low intensity. We need to use powerful lasers in order to get most of the information that we get out of the particles as they flow for a limited time across the laser beam. Yeah. So the way I picture this typically is, imagine you're taking a picture of something that is around 10 microns, that is flowing at 20 meters per second across the camera, essentially in front of the camera, but you only have a few microseconds of exposure time. So imagine how sensitive the camera has to be, or how powerful the light source needs to be in order for the image to be actually resolved from electronic noise. And in cytometry, the technology have been evolving over time. So the principle haven't changed much, but what have changed a lot is the number of lasers as they become available, the power they have, and the number of detectors that you have. So this is an example of a chronology of development in cytometry from one laser and two colors at the early stages of the technology, up to 7 to 10 lasers and 50 detectors in the case of the Symphony instrument from Becton Dickinson. All of this following a kind of one detector, one fluorochrome approach. So in the traditional instrument, every single detector is dedicated to the measurement of a specific fluorochrome. Yeah. Now, in recent years, with the arrival of the spectral analyzers like the Aurora, well, you can have instruments that have 5 lasers, 64 fluorescent detectors, and measure up to 45 markers, which is absolutely insane. Like 2 years ago, no one thought it was possible to put so many fluorochromes and measure them independently from each other in a single cell and be able to measure something. But this is the state that we are at the moment. This is a beautiful advantage and that comes with the spectra, a mixing of fluorochromes instead of the traditional one detector, one fluorochrome approach. And we have several of these instruments in the platform as well. Now, what is measured in cytometry? One thing is light scatter, and the reason why light scatter is so important, well, I will explain you in the next slide. Scatter defines what is 100%. Imagine if you're trying to measure GFP, the percentage of GFP expression in a cell line, you need to know how many cells there were in total, regardless of fluorescent content. So scatter becomes a kind of a way to identify all the events that matter in the sample, and on these events you can, for instance, measure how many of them have fluorescence. So, and scatter is very useful because it's a very generic property. It happens in many different ways and shapes and forms, and most particles will scatter anyway. So you can always get this idea of the 100%. And although scatter is produced across all angles. Yeah. Cytometry uses most of the instruments. I mean, there are variations between instruments, but most of the instruments use two components of the scatter that is produced. One is called the forward scatter. It's called forward because it's measured roughly in the direction of the incoming light source, or the laser in this case. So it's a scatter that is produced around the main laser path. That's called forward scatter. And the other one is a side scatter, or the measurement of scatter that is produced at 90 degrees from the incoming light source. And the reason why forward scatter and side scatter are used is because they allow you to get two measurements that are independent measurements of scatter that have slight reference to properties, general properties of the particles. Most noticeable in the case of immunology, things like size of the particles. In this case, forward scatter tend to be considered to correlate with the size of the particle, the size of the cell, and the side scatter tend to correlate with the complexity, the inner complexity of the cells and the membranes. So you can think of it as two measurements of scatter that combine give you an idea of diversity within a sample. And the typical plot, and I think the classical plot in terms of scatter, forward and side scatter measurements and how they correlate with properties of the cells, is what you see when you run, for instance, blood or lyse blood through a cytometer. So you have distinctions between erythrocytes, which is the gate in red, you have lymphocytes, then you have monocytes and granulocytes. And as they increase in size and complexity, well, they increase in forward and side scatter. And you can see, of course, the different particles, with different cells within the whole leukocyte and hematopoietic path or differentiation will have different properties, and these properties will correlate with scatter measurements. But something that you need to remember, because it's extremely important, that unless you're comparing the same type of cells, in this case leukocytes, or cells within the same subclass like lymphocytes and monocytes, activated, differentiated, proliferating, etc. This correlation with complexity for the side scatter and size for the forward scatter do not always apply. And it is very important to understand that scatter is a little more complex than these correlations, and specifically if you're working in nanoparticle studies, this is crucial. Nanoparticles and microbiology. Now, thinking of scatter is a broader view, and the broader power, I'm putting this example that we all know of how the scatter of X-rays into a crystal of DNA allows the discovery of the double helix structure, which is a Nobel Prize in biology. Now, this is scatter as well, and the correlation is not that strict between size or complexity. It had to do with the molecular components of a given crystal within a given crystal and the way they are arranged in space. And scatter is a kind of generic term that we use for things that are that we used to see all the time. So one of the things is diffraction patterns. This is scatter, a different kind of way of scatter. You have refraction, you have reflection, and you also have interference. So if you consider light of waves, and you will have interference between the waves that are generated. So when you're thinking of scatter and the laser hitting the particles on the cell, you have to imagine that every molecule within the cell, every component within the cell will interacting with the light in a way that will produce reflection, refraction, and diffraction. Combined via interference patterns, they will produce main outcomes that can be measured as forward scatter, side scatter. Do they relate with size and complexity? Maybe. Sometimes they do. But in some cases they won't. And when you consider the size that typically can be detected in cytometry, if you consider the scale of all things, so I got this from Wikipedia, most cytometers stop right at the 200 nanometer particle size in terms of scatter definition. Yeah? They have 200 nanometers as a limit in terms of the minimal size that they can detect. Most instruments. Instruments like the Union Biometrica biosorter can measure whole embryos and small organisms. But we don't have any of these. So most instruments in our case will go all the way from 200 nanometers all the way to macrophages, neurons, things like that. Some instruments like the Cytoflex that we have and the Astro CQ that we have can measure 80 nanometers, down to 80 nanometers. So this is a major jump, and I will explain this later, but is something that is a testament to the sensitivity of the reflex and the ability to align the astros in a way that you can reach these kind of benchmarks. And I'm not promising anything, but I know that several companies are working on extending this detection even further, so that they're trying to achieve detections on the order of 30 nanometers, which is roughly the size of viruses. Yeah, and all this development and this push for lower and lower sizes being detected by instrument have to do with the exosome field and all the results and the projects that are working with exosomes that are typically below 200 nanometers, and they are difficult to measure in a conventional instrument that have been traditionally designed for lymphocyte and bigger cells. Now, scatter as a whole, and this is a kind of a software that allows you to predict scatter patterns, depends on several things. It depends, for instance, on the size of the particle, yes, in this case, sphere and particles. It also depends on the refraction index, which reflects on the content and the nature of the particle itself. Not only the refraction index of the particle, but also the refraction index of the medium. Yeah, for instance, if you put a little ball of glass into water, you will not be able to see it. And the reason why you don't see it is because the refraction index of the water is slightly close to the material that makes the beads, yeah, these glass beads, which means that there will not be bending of the light. You will not be able to see the shape. You will not be able to distinguish the sphere from the surrounding liquid. So refraction index ratio is very important. Laser wavelength is also very important. So in this case, 488 nanometers, that's what I use for this model, but it's a laser that is mostly used for scatter. But also depends on the orientation of the laser, incoming laser wave. So if it's polarized in a vertical manner or horizontal manner, the patterns of scatter that you will get will change. And you can see that based on these calculations, so a particle roughly 200 nanometers using 488 and the refraction index of the particles and the media typically used in the instrument. This particle, I think, is polystyrene. You can see that the scatter tend to be mostly in the forward direction, yeah, very little in the side scatter direction, and that's why most instruments use a high efficient photon detector for the side scatter measurements, and forward scatter can be measured with things like photodiodes, which are less efficient. But it's a complex function, that's what I mean. And as you decrease the size of the particle, in this case I'm applying the same model that I showed you before to go from 1 micron all the way to 50 nanometers. You can see that not only the intensity changes, but the pattern of the scatter and the distribution, the angular distribution of the scatter changes. So from the typical high forward scatter, low forward scatter kind of arrangement that you see in things like lymphocytes, you get all the way to 100 nanometers with a pattern of scatter production that is seemingly circular. So there is no major difference between forward and side scatter, and the lights as a whole have been reduced in intensity quite a lot. And I'm showing you this here in this example. So when you go from 200 nanometers to 100 nanometers, this amount of forward scatter light that you're producing with 200 and with 100, this is roughly 100 times less light when you have the size of the particle. And the reason of this jump, so this 100 times less light produced by 100 nanometers compared to 200 nanometers, explain why most instruments stop at 200. So going from 200 to 100 in terms of scatter measurement represent a big jump in terms of signal decrease reduction. And you need to have instruments that can cope with that, can be able to measure that efficiently. But not only the intensity of the particle changes, the noise is overwhelming. So if you imagine, if you consider what scatter is measuring the light in the same wavelength of the laser. So these are the same kind of photons with the same kind of wavelength. The challenge in scatter measurement is how do you measure a particle from a surrounding overabundance of laser photons that are coming to hit the particle. So you need to have some special optics to differentiate nanoparticle signal from the noise that is coming from the laser. And these are two examples that I can show you here in the platforms. In the case of the Fluidics EQ, you can see the scatter patterns projected on the side view, and you can see all the diffraction and interference patterns that are generated towards when the laser encounters the particle and all the way to the forward direction. And the cuvette cases, in the case of the Forteza, in which you see light going everywhere, the amount of noise is kind of impressive. And noise is as important when it comes to resolution of signals as the quality of the lenses and the sensitivity of this instrument as a whole. And I think this is a perfect example to understand why, how important noise is. Imagine if you were living in a city center, the amount of light pollution that you get will stop you from being able to see the Milky Way and the stars. Only when you go progressively out of the cities into perfectly isolated areas with no light pollution, your eyes, which are the same tools to see, highly sensitive, can see now suddenly the Milky Way. So background can mask and essentially prevent the detection of dim signals, regardless of the detection system, of the sensitivity and the quality of it. So just to show an example of how instruments handle scatter of different sizes and particles, I'm showing you examples of the Cytoflex S and the LX at the dental school to show you how Cytoflex can measure down to 80 nanometers polystyrene beads. So in terms of exosomes, we were talking about maybe 100 nanometer, maybe it can measure something like that. So yeah, these instruments, which are exquisitely sensitive, the Cytoflex instruments, not only are sensitive in the fluorescence side of things, but also in the scatter view. A few years ago, we ran this experiment on the Astros, the Moflo Astros, which is a jet inertial sorter, and the aim of the experiment will to optimize the scatter alignment for the forward assembly, which is quite complicated and fancy in the Astros, with a lot of fine-tuning that you can apply in terms of masks to block different components of the noise. The BSO, which is the beam shaping optics, is the optical elements that will shape the beam and will focus the beam right where the particle is flowing and also aligning the jet. So it was a kind of multi-aligning procedure that typically will take like 20 steps to try to find the limit of detection in terms of scatter of an instrument like the Astros. And the surprise for us is that although the company will tell you that the limit of detection of the Astros is 200 nanometers, we managed via this successful sequential alignment process to get to a full resolution of 200 from the noise, roughly 100 times less noise measure than the scatter that was produced by the 200 nanometer beads that we were using. And by running different beads that have essentially precise size definition, different materials like silicon or polystyrene, we managed to accommodate lots of bits between the 200 and the noise. In our case, we managed to see that the instrument was able to see down to 80 nanometers, clearly resolved from the noise. Now, the effort that we invested into this kind of alignment, it shows you how critical alignment could be in terms of controlling the noise, increasing the sensitivity of the instruments, and of course it explains the limits that we have in terms of scatter detection. Although you can reach these kind of detection limits like 18 nanometers in the Astros, the effort and the number of steps that you have to follow through to get to there is excruciating. But it's something that is possible, and it was as good as an exercise. Now, fluorescence is the most important measurement that is done in cytometry, at least in the instruments that we have in the platform. And fluorescence, again, happen in all angles. Instruments tend to collect fluorescence that is produced in a section of the fluorescent sphere, roughly in the same position that the size scatter is collected. And why fluorescence? Well, I think Ellie did a pretty much impressive introduction to fluorescence in the last lecture, so I will not be going into full details. But one of the reasons fluorescence is used is that at the speed of the interrogation that I mentioned before, from milliseconds to microseconds, you need to have fluorescence because it's extremely quick. So from the moment the particles are excited to the moment the emission happens is essentially extremely quick, in the order of nanoseconds. And that would explain how a particle traveling the laser will produce not only one fluorescent cycle, but several cycles of fluorescence. Now, the speed of the interrogation do not lead to, in most cases, to bleaching. So we don't have photo bleaching as a main problem in cytometry. It clearly depends on the dye, but most of the times dyes across the laser are excited several times, and they can be reanalyzed, and the fluorescence is still there. But important things about fluorescence and understanding fluorescence is that there is an excitation maximum, which is the wavelength that produces most likely the biggest excitation of the dye, the increasing the probability of excitation. And you also have the emission maximum, which is where the photons most likely will be produced in terms of probabilistic emission. And there is a difference, of course, between the laser of excitation and the emission that you get. The difference here is the stock shift. Now, the lights that we're talking about here, the fluorescence, is just a section of the visible light. It goes all the way from a little bit above UV, that's what instruments can measure, all the way up to infrared. So this is the fluorescence that typically we collect. And there are many types of dyes. There are dyes that are essentially organic. For instance, it can be fluorescent proteins, of course, or it can be pigments contained within fluorescent proteins. You have also GFP as a classical example of fluorescent protein. You have small synthetic molecules like FITC. All of them organic. This is a common trend between all of them. You have the tandem dyes, which are essentially two dyes coupled together that work a little bit like a thread pair. And you got, of course, polymers and things that have recently developed, like the brilliant violet dyes from Becton Dickinson. Now, in cytometry, the aim is to measure as many fluorochromes as possible, and sometimes after exciting with a single laser. And I'm showing you here an example of maybe one of the earliest configurations for detection in cytometry: excitation with a 488 nanometer laser and emission of FITC, PE, PERCP size 5.5, and PE size 7, for example. And you can see that although each of the dyes and the design of the instruments are meant to capture the fluorochromes, each of the fluorochromes at the maximum of emission, well, fluorochromes also emit in different regions. And the first question for us is, how do you capture the maximum? So that is handled by the optical design of the instrument, and specifically the filters that are in place in cytometers to essentially dissect the spectrum into components that will end up with the capturing of the maximum information of the specific dyes. And there are three main types of dyes in cytometry. You have what is called as SP, or dichroic short pass filters, that allows only wavelengths that are under a given wavelength to pass through the filter, and the rest is reflected. You have the dichroic long pass, so it's similar to this, but in this case the light that goes over a given wavelength is transmitted through the filter, the rest is reflected. And then you have the bandpass, which is the most important filter that you guys need to know, because it's a filter that is placed right before the detector, and it will enable the collection of wavelengths within a specific range. So 530-40 means 530 center wavelength, 40 nanometers the total bandwidth of the filter, which means that the amount of light that will be collected, or the definition of the wavelength that will be collected by this filter, will be 530 plus minus 20 nanometers, half the bandwidth. And different instruments will have different configurations. In this case of the Satoflex, where you have different bandpasses, and most of our instruments will have these kind of tables in front of the instrument, so you can see which detector are in place, which bandpasses have been used, and which are the fluorochromes that you can analyze in the instrument. And the design have been changing over time, from less efficient systems that are extremely complicated, combining long passes, short passes, band passes, to split the light into different shapes and forms. And in this case, I'm showing you an example of a panel that has a fluorochrome that will not be acquired by any of the band passes. So knowing the band pass, knowing the emission maximum of the fluorochrome is important in order to make sure that the measurement can take place. Going to the instruments like the Fortezas, which are more efficient than the previous configuration, in which you combine reflections and band passes and long passes to essentially split the spectrum of light into different components, and photons are typically acquired in the reverse order of the wavelength. So higher wavelength, lower energy photons are collected first, they are less efficient for the PMTs, and so on. You go all the way down to very close to the laser. So the last example I have here of way the optics of an instrument is arranged is the example of the Cytoflex and the Aurora. Now, technology have been evolving over time, engineers are smarter and smarter, and the acknowledgement of the limitations in terms of quantum efficiency of instrument, in terms of the ability and the efficiency of photon collection, and the concern for it have been increasing. And in the case of the Cytoflex and Aurora, you have an example of probably the most efficient way of collection at this point in time. So it's a system that combines bandpasses and highly efficient mirrors that reflect lights in all wavelengths. So the light essentially arrives into this array of filters, hit the first bandpass. If there is light within this bandwidth, it will get into the detector. Light that is not within the bandwidth will be reflected into the mirror, sent into the next detector, and so on. So you get a combination of reflection, which is highly efficient in terms of optical quantum efficiency of photon translations, and the bandpass that will filter the light to measure each of the spectral components. So now, once the light have been handled and dissected into spectral components, as I mentioned, depending on the fluorescence that you want to measure and the instrument configuration, you arrive at the photon detectors. And probably the best example of a photon detector that have been in the field for years and years and is still around is the photomultiplier tube. So you need to have a very sensitive kind of detector to handle the dim signals that are coming out of the interrogation point where the laser meets a particle at the flow cell. And in the case of the PMT, what you have is a series of amplification steps that happen once the photon hits the PMT and meets the cathode. You get electrons that are produced out of the interaction between the incoming photons and the materials making the photocathode, and these electrons will jump through a cascade that will sequentially amplify further and further the signal, all the way up to 10 to the 8 times the incoming number of photons that arrive to the PMT. And it's extremely important because it will allow to measure something out of a signal that is extremely dim. Another type of photon detector at this point that we also have in the platform is the avalanche photodiodes, which are used by the Aurora and the Cytoflex. And these are a different operation. The design is different, the principles of operation is different, but the major take-home message here for these detectors is how sensitive they are. So this is a graph of the quantum efficiency of the avalanche photodiodes, the APDs, compared to the PMTs. And where in the PMTs you can see that for higher wavelengths the quantum efficiency decreases, and the maximum the PMT can reach is something like 0.2, around 0.2, which means that 4 in 5 photons received by a PMT are wasted and only 1 photon makes it into the signal. The APDs tend to be very much consistent in terms of the quantum efficiency, much larger than the PMT, so the 0.8, and across the whole wavelength range, which explains why these instruments are so sensitive in regions or measurements of fluorescence in the infrared, in the red spectrum, and the instrument essentially helps a lot with the sensitivity limits of the instrument as a whole.